Bismillahirrahmanirrahim. Here's Malaika Adib from Chemistry Department. The topic of my today's presentation is removal of heavy metals by using plants. There are some contents. Let's have a look. Heavy metals introduction, their sources and effects, methods for removal of heavy metals, rhizofiltration, plants that use for rhizofiltration. Is wastewater necessary for growth of rhizofiltration plants? Specification of rhizofiltration plants in their function, data requirements for rhizofiltration, function of plants for aquatic treatment, uptake of metals by plant roots, its construction, working model of rhizofiltration, why we use rhizofiltration plants for treatment of wastewater, metal adsorption analysis, cost, applications, advantages, disadvantages, phytoextraction, phytovolatilization, phytostabilization, and conclusion. First of all, the introduction to heavy metal. Heavy metals are natural elements characterized by their atomic mass, and their atomic mass in periodic table starts from 21 to 31, 39 to 50, and 72 to 83. Let's have a short introduction of periodic table elements according to their atomic mass. Like metallites are present in periodic table with their atomic mass 5, 14, 32, 33, 51, 52, and 84. Metals are present on the right side of the periodic table and non-metals are present on the left side of the periodic table. Although heavy metals occurring in low concentration, they can be found all through the earth crust of our planet. Commonly, a density of 5 gram per cubic centimeter is used to define a heavy metal and to differentiate it from other light metals like sodium, lithium, magnesium, aluminium, potassium, calcium, and beryllium are known as lightest metals. Moving to the next slide. Some heavy metals like copper, selenium, or zinc are essential trace elements for various biological processes, also driving the entire human metabolism. Like cobalt, acting as central atom in the vitamin B12 complex, it is a key player in the reductive branch of the propanoic acid fermentation pathway. On the other hand, many of these, like chromium, nickel, cadmium, arsenic, etc., classically represent the dark side of the chemistry. They exert toxic effect on environment even at low concentration. Moving to the next slide, there are sources of heavy metals, anthropogenic sources and natural sources. If we discuss about anthropogenic sources, arsenic is produced from pesticides, fungicides, metal smelters. Cadmium is produced from welding, electroplating, batteries, nuclear fission plant. Chromium is released by mining, textile, electroplating, tannery industries. Copper is released from electroplating, pesticides, mining. Lead is produced by paint, batteries, automobiles, emission, burning of coal. Manganese is released from welding, fuel addition, ferromanganese production. Mercury is released from pesticides, paper industries, and batteries. And nickel is released by electroplating, zinc based casting. And natural sources are volcanic eruption, weathering of rocks, natural forest fire, particles released by vegetation. Moving to the next slide. In this slide, we discuss about the effects of heavy metals on our environment. In this picture, we can see the heavy metals are produced by burning of forest, volcanic eruption, leaching, industrial discharge, pesticides and fertilizers, and electronic waste. These heavy metals are arsenic, mercury, lead, cadmium, chromium, and copper, etc. These all heavy metals exert toxic effects on our environment directly or indirectly. It pollutes our aquatic system. It also contaminate and destroy our land, our crops, and ultimately these all exert hazardous effects on our human health. If we discuss about the effects of heavy metals on the aquatic system, in aquatic system they can cause eutrophication, disturbance of aquatic ecology, and deposition of heavy metals in fishes. The heavy metals accumulated in the gills of the fishes and become difficult to breathe. And due to the accumulation of heavy metals in the in the gills of the fishes, so they can die. It also contaminate our groundwater and it cannot be used for drinkable purposes. If we discuss the effects of heavy metals on our land, it decreased the soil health, altered the microbial biomass and activities. It caused the abnormal nitrogen transformation and irregular carbon mineralization. And due to the disturbance of land, it also affect our food. It caused the aberrant genetic response, oxidative stress, improper fertility responsiveness, change in signaling, and biosynthesis pathway. These all affect the human health, like is it cause the mutagenicity, carcinogenicity, genotoxicity, neurotoxicity, endocrine disruption, and it affect the human health even at low concentration. Moving to the next slide, there are the methods for removal of heavy metals. In the previous slides, we discussed how metals are produced, from which sources they are produced, and how they affect our whole environment. Now we see by using plants how we can remove these heavy metals from our environment, from our contaminated water and soil. Basically, phytoremediation, phyto means plants and remediation means restore balance, is a technique in which green plants are used to remove, detoxify, and decompose hazardous metals from our waste water or waste soil. For soil and water contamination, some types of phytoremediation methods are used, like phytostabilization, phytoextraction, phytodegradation, phytovolatilization, and rhizofiltration. Moving to the next slide, in this picture we can show how the plants uptake heavy metals from the contaminated side. First of all, we see the uptake pathway in roots. There are two pathways, apoplast pathway and symplast pathway. The blue arrows show the symplast pathway and the yellow arrows show the apoplast pathway. And these are the capsarian strips which are present on both sides, and these capsarian strips block the apoplast pathway. So heavy metals does not move further. The heavy metals moves through the symplast pathway and heavy metals translocate to shoots through simplest pathway. In the next picture, we can see the xylem vessels in shoot means the movement of heavy metals from roots to the shoots and then further. In shoot we can see the upward movement of the heavy metals in the different colors of heavy metals show the different types of heavy metals like zinc, copper, iron, cadmium, lead, etc. And these arrows show the upward flow of heavy metals, and at the end xylem unfolding of heavy metals. In the next picture, we can see the movement of heavy metals in the leaf cell vacuole. In the middle of the picture, it is the vacuole, and the pink area of the cell vacuole shows the plasma membrane. These heavy metals stored in the vacuole, like in this picture, we can see the cadmium, zinc, copper, nickel, and lead present in the vacuole of the leaf. These move through the cell wall of the leaf and then present in the vacuole. The green area shows the cell wall, and there are two dots or two circles present, pink and blue. These are the heavy metals sequestered in the cell wall. In this picture, we can see how the in which pathways moving from roots to the shoots and then to the cell vacuole. Moving to the next slide. Basically, the topic of today's presentation is the removal of heavy metals from wastewater. And rhizofiltration is there are many other techniques uh, which use plants to remove heavy metals, but we want to use heavy metals from wastewater. So rhizofiltration is the technique which is used to remove heavy metals by using plants from our wastewater or contaminated water. Rhizofiltration is the adsorption onto the plant roots or absorption into the plant roots of contaminants that are in solution surrounding the root zone, and the surrounding root zone is called rhizosphere. It is a phytoremediation technique that is related to phytoextraction or phytoaccumulation. Means in phytoextraction or phytoaccumulation, we can also use plants for the removal of heavy metals. But in rhizofiltration, we remove heavy metals from wastewater. But in phytoextraction or accumulation, we remove heavy metals from contaminated soil. It is basically used to remove the heavy metals like mercury, cadmium, copper, zinc, and arsenic, etc., from contaminated wastewater. Moving to the next slide, which plant species in this slide we discuss which plant species are used for rhizofiltration. A few common species that used for rhizofiltration are sunflower, tobacco, and in these pictures we can also see how these plants are constructed, and in which pathways the heavy metals move from roots to the shoots and then leaves. Rye plant, Indian mustard plant, and in these pictures also we can see the movement of heavy metals. Spinach, and corn. These are the some species which we used for rhizofiltration. Moving to the next slide, is wastewater necessary for plant growth? In this slide, we discuss that it is necessary that we grow the plants that we used for the removal of heavy metals in wastewater. Means, can we grow these plants in clean water or not? Let's see. The rhizofiltration technique can be constructed either as floating crafts on ponds or as tank systems. The plants are first grown in clean water and after they transferred to the contaminated site to remove heavy metals from wastewater. It means we grow these plants first in clean water and then move to the contaminated side. Rhizofiltration involves growing plants in a greenhouse first and then transferring them to the remediation side. Great care must be taken to maintain an optimum pH in the effluent solution. Moving to the next slide. In this slide, we discuss specification of rhizofiltration plants in their function. In this slide, we basically discuss is rhizofiltration plant is specific in function to remove a specific heavy metal. The rhizofiltration plants which are used are specific in their function to remove heavy metals from wastewater. It means each heavy metal is removed by using a specific rhizofiltration plant, such as P. australis could be used in rhizofiltration system to deal with pollution related to nitrogen species in warmer months. Nitrates were removed up to 60.4% by plants, while in systems without P. australis, only 45.4% was denitrified. Aerodonics constructed wetland could deal with copper pollution, rhizofiltering the pollutant, offering in 30 days remaining concentration of 3.5% of the initial copper in water. Moving to the next slide. Ischia stratiotes proved to be a phytoremediator to rhizofiltration of polluted Egyptian wetlands. It possesses an interesting potential of rhizofiltration for chromium, lead, and copper, and 10 times higher for iron. Barley, oat, pea, corn, and timothy in bioplater using foam plastic as substrate could remediate radionuclides such as cesium-137 from polluted water. This system offered decrease of 80% in radioactivity in 24 hours, 90% in 48 hours, and 97% in 5 days, mainly due to Timothy's promoted rhizofiltration. It means to remove a specific heavy metal from contaminated site, we use a specific species of plant. Moving to the next slide, data requirements for aquatic contaminants. Depth of contaminants, types of heavy metals present, level of contaminant must be determined and monitored. Vegetation should be aquatic, emergent or submergent plants. Hydraulic detention time and sorption by plant roots must be considered for a successful design. Moving to the next slide, functions of plants for aquatic treatment. It means the different parts of the plants plays a specific role in the cleaning of contaminations from aquatic treatment. Roots or stem in water column, uptake of pollutants used for surface on which bacteria grow, and media for filtration and absorption of soil, and stem or leaves at or above water surface attenuate sunlight, thus can prevent growth of algae, reduce effects of wind on water, and reduce transfer of gases and heat between atmosphere and water. Moving to the next slide. In this slide, we discuss the uptake of metals by plant roots. The plants which are used in this technique are initially raised hydroponically and later transferred to the contaminated water source after having developed an extensive root area. Such plants are harvested when the roots become saturated with contaminants. Shoots can be discarded or allowed to regenerate a new root depending on the species which we used for. Dried material may be combusted to further reduce its mass and to produce energy for the operation of rhizofiltration system. Moving to the next slide. Acids may be used to strip metals from the plant residue followed by reclamation of metal from the acid solution. Alternatively, metal enriched plant material can be buried at the hazardous waste site. In this method, both aquatic and terrestrial plants are used. However, terrestrial plants are preferred owing to their fibrous and long roots. Moving to the next slide. This slide shows the construction of the model of the rhizofiltration. Hydroponic units consist of a PVC plastic cylinder which are 12 centimeter tall and 10 centimeter in diameter. It contains four plants supported by a metallic grid positioned 7 centimeter from bottom and 10 centimeter deep layer of volumiculate placed on top of grid contain 4 liter of nutrient solution. In this picture we can see the how these plants are grown in the aquatic system and the hydroponic cylinder. Moving to the next slide. In this slide we discuss the working model of the rhizofiltration, how these plants are constructed. We can see in this picture the all in this picture we can see all the rhizofiltration units. There are four rhizofiltration units which are interlinked through the pipelining, and these pipelinings are used for the movement of contaminated water from one side to the other, and these arrows show the movement of the water from one pipe to the other, and from one filtration unit to the other. And there are pumps are present to pump the contaminated water from one side of the pipe to the other side and from one unit to the other unit. First of all, the contaminated water with toxic metal is pumped from the first pump, which is present on the left side, and and it moves through the through all the radiofiltration units and with the proper pH and temperature adjustment, and at the end all the toxic metals removed, and it is present in the filters, the water present in the filters, and at the end if any species left, it is removed by using the filters and clean water is removed. Moving to the next slide. Why we use these plants? There is a question that there are many other land plants. Why we not use these plants for Rhizofiltration for the removal of contaminants from wastewater. Why we use specific plants? It means these specific plants have some special qualities. That's why these are used for rhizofiltration. That's why these are used for the removal of waste, for the removal of heavy metals from wastewater or from waste contaminated site. Let's see. The roots of these plants have an intrinsic ability to absorb and precipitate heavy metals from solutions. The roots of these plants are genetically modified and are specific in their action to remove heavy metals. The roots of these plants use several mechanisms for metal removal, and these mechanisms are not specifically similar for different metals. These mechanisms may be physical, chemical, and biological. Such physical and chemical processes are chelation, ion exchange, and chemical precipitation of metal ions. Biological processes are probably responsible for the slower components of metal removal from the solution. These biological processes include intracellular uptake and translocation to the shoots. Fortunately, the ability of plants to translocate lead to shoots varies much more than their ability to accumulate metals in roots. These are the specific properties due to these we use specific plants for the removal of heavy metals. Moving to the next slide, metal adsorption analysis. We know plants are used to remove heavy metals from the contaminated side. In this slide, we basically see uh, at which concentration which plant remove heavy metal from contaminated side, and it is different in different plants, and we can measure it by using a spectrophotometer. Heavy metals in root tissue were analyzed by using a fission direct current plasma spectrometer DSP model SS7. In this process, roots were dried for 2 days at 80 degrees Celsius, ashed at 500 degrees Celsius for 6 hours. The ash was dissolved in 2 parts of 1 molar nitric acid. Lead concentration in solution were analyzed directly using a Perkin Almir 603 atomic absorption spectrophotometer, but all other metals in solution were analyzed by DSP, means direct current plasma spectrometer. Moving to the next slide, cost. In this slide, we discuss if we construct a rhizofiltration unit, how much it cost, means it is cost effective or not. Rhizofiltration is a cost effective for large volumes of water having low concentration of contaminants that are subjected to stringent strength. It is really inexpensive yet potentially more effective than comparable technologies. The removal of radionuclides from water using sunflower was estimated to cost between $2 and $6 per thousand gallons of water treated, including waste disposal and capital costs. Moving to the next slide, there are applications of rhizofiltration technique. Rhizofiltration may be applicable to the treatment of surface and groundwater, industrial and residential effluents, agriculture runoffs, and other contaminated solutions. Plants suitable for rhizofiltration applications can efficiently remove toxic metals like lead, cadmium, chromium, mercury, zinc, and nickel, etc., from a solution using rapid growth root systems. It also removes the low level radioactive contaminants from liquid streams. Low level radioactive contaminants originate from many sources such as the nuclear power industries, medical and academic institutions, and a host of industries that use small amounts of radionuclides in their manufacturing process. Reprocessing of nuclear fuels also creates additional low level radioactive waste. These which generally do not pose severe risk from their radiation fields, but there is a risk associated with nuclides migrating through the environment and entering the food chain. These plants can grow on land of marginal quality and have long lifespans. For decontamination, hybrid poplars with roots extending 30 feet deep have been used. Moving to the next slide. Advantages. Rhizofiltration is a contamination treatment method that may be conducted in situ, with plants being grown directly in the contaminated water body, or ex situ, where plants are grown offsite and later introduced to the water body. In some cases, contaminants may have been shown to be significantly decreased in a very short amount of time. One study found that roots of sunflower reduce level of uranium by nearly 95% in just 24 hours. After harvesting, the crop must be converted to biofuel, briquette, a substitute for fossil fuels. Moving to the next slide, disadvantages. This contamination method has some limits. Any contamination below the rooting depth will not be extracted. The plants that are used may not be able to grow in high contaminated areas. It can take years to reach regulatory levels. This results in a long time maintenance. Plants grown on polluted water become a potential threat to human and animal health, and therefore careful attention must be paid to the harvesting process, and only non-furrow crop should be chosen for rhizofiltration remediation method. Moving to the next slide. In these slides, we discuss the other methods that use plants for the removal of heavy metals, not from wastewater, from contaminated soil. Phytoextraction or phytoaccumulation is the uptake of contaminants by plant roots and movement of these to the above parts of the plants by absorbing, concentrating, and precipitating the contaminants. In this method, hyperaccumulators have tendency to taken up non-essential elements 100 times greater folds by non-hyperaccumulators. For the plants to take up heavy metal contaminants, the contaminants need to be converted into water soluble compounds. This technique is called induced phytoextraction. Moving to the next slide. For the plants to take up heavy metal contaminants, the contaminants need to be converted into water soluble compounds. This technique is called induced phytoextraction. Metals and allow easy uptake by roots. In this technique, chelating agents are added into the soil that dissolve the toxic metals and easy uptake by roots. Moving to the next slide. Phytovolatilization. Phytovolatilization involves the uptake of contaminants by plant roots and its conversion to a gaseous state and release into the atmosphere. In this picture, we can also see the heavy metals move from roots to the shoots and then to the leaves, and after that these released into the atmosphere. This process, driven by the evapotranspiration of plants, mercury and selenium can be removed in this way. Phytostabilization. Phytostabilization is the use of metal tolerant plant species to immobilize the heavy metals below ground and decrease their bioavailability, thereby preventing migration into the ecosystem and reducing the likelihood of metals entering into the food chain. It mainly focuses on sequestering pollutants in soil near the roots but not in the plant tissues. At the end conclusion, in this presentation we discussed the rhizofiltration method for the removal of heavy metals, and it is an efficient, cost effective and eco-friendly technique to deal with the pollution of the environment which is caused by various sources, and it plays an important role for the removal of heavy metals from contaminated sites by using in situ or ex situ process. Thank you.